Welcome back. Now we're going to get into actually how action potentials work. So this is another one of those kind of big physiological processes. So you're going to want to break this down into its component parts. It will help out quite a bit. So we're going to talk about potentials, membrane potentials. So neurons, and we learned about nervous tissue and said that it could both generate and conduct electrical signals, impulses. So basically, if a neuron is stimulated enough, that's how we get it to generate an electrical impulse that is then going to be conducted all the way down its axon, where it will then communicate with the next cell in the line. So this impulse that we're talking about is called an action potential, or what the layperson would call a nerve impulse. So let's talk a little bit of physics here for a second. The body is neutral, but there are definitely areas where one charge predominates over the other. Okay, so overall, we're not walking around carrying a charge, but there are certainly places where the charges are not distributed equally. Now, whenever you have that, an unequal distribution of charges, particularly a separated cross a membrane, there's potential energy in there because those charges are attracted to each other. Voltage is a measure of that potential energy. So we use voltage to determine how much potential energy is in a system. And then that possible energy is called the potential. So think about like a 9-volt battery. That's how much energy, potential energy, is in the battery. But you can't just use it until you connect it to something and you allow there to be a flow of the ions, and that's what a current is. So when we talk about the flowing of sodium across the membrane, the flowing of potassium across the membrane, that's a current. It's an electrical current that is occurring. Where we're allowing charges to flow generally down their gradients. So we know that the membrane is not permeable to ions. We learned that. So then the question is, how is this flow occurring? And we're going to have a bunch of different types of channels involved. Leakage channels are pretty much exactly what they say. They leak ions. We're not really going to get into them because they are not principal components of action potentials, but ligand-gated and voltage-gated channels absolutely play a major role in action potentials. Now, what we're going to be flowing down are what are called electrochemical gradients, meaning that there is an electrical component to the gradient, the charges, positive attracted to negative, etc., and a chemical, which is a concentration gradient. So both of those are going to come into play in terms of how ions move and how efficiently they move. All right, let's talk about our two types of channels. Ligand-gated, this one's called chemically gated, but it's ligand-gated, is going to be a channel in the membrane that requires you to bind a specific ligand to open. In terms of our neurons, our ligands are neurotransmitters. So you guys saw really good examples of ligand-gated channels when you were studying muscle contraction. You bound your acetylcholine and the channel opened and sodium came in. That is an example of a ligand-gated channel. Now, you can picture that the level of response in terms of ligand-gated channels is going to be directly proportional to the concentration of the neurotransmitter or the ligand. So ligand-gated channels are going to give us what are called graded responses. The more ligand I have, the more channels I can open, the more ions move, the bigger the response. Okay, so you just picture when you're at Meijer right after work, peak time, there's tons of people, there's a huge gradient. There's a bunch of people waiting to check out and not that many people who have checked out. So if they begin to open up more cashiers, more people are going to be able to get through the lines. a lot faster. So the more cashiers are open, easier it is to get through the lines. Here, the more ligand gated channels are open, the more ions can flow, and that's going to be directly related to how much of the neurotransmitter or the ligand I have. Now, voltage gated channels don't require any sort of ligand to open them. They open as soon as the membrane potential hits a certain voltage. So that's their key. These guys are going to give us what are called all or nothing responses. They are not graded at all, because when you hit the voltage, all of them open fully and completely at the same time. So picture you've got a toll booth. So you got to pay money to get through the toll booth, and the toll is 50 cents. Okay. If you put in 49 cents, it doesn't open. If you put in 50 cents, it opens completely. If you put in 51, it doesn't open more. It's already completely open. So that's an all or nothing response. You either hit your 50 cents and it opens 100% completely open, or you don't, and it doesn't open at all. At 49 cents, it doesn't open 90% of the way. Okay, so you've got an all or nothing response when it comes to a voltage gated channel versus a graded response when it comes to a ligand gated channel. So let's talk about resting membrane potential. So what this means is when there's nothing stimulating the neuron, it's not busy doing anything, the potential is negative 70 millivolts. So here we have our neuron, we are measuring it, there's nothing going on in the neuron. The difference between the interior and exterior is measured to be negative 70. At this point, we say that the cell is polarized because it's different from one side to the other. The interior of the cell tends to be more negative. We see all of our little negative signs inside of here because we've got DNA and we've got proteins that are both generally negatively charged, and they are stuck inside the cell because they are very, very big and cannot diffuse out. So we tend to see that the interior is more negative. The major intracellular cation is potassium. We have a lot of potassium in the intracellular fluid. The exterior tends to be more positively charged when we compare it. Major extracellular cation is sodium. Major extracellular anion is chloride. Now, at the resting state, the membrane is more permeable to potassium than it is to sodium, so that's where those leakage channels come in. So we have the sodium-potassium pump that maintains the gradient so that we always have more sodium outside and more potassium inside. All right, so let's talk about how we change the potential. If resting membrane potential is 70 and that's in a neuron where nothing's happening, we need to figure out how does it change, because that's going to be where we're going to get the functionality here. So we can depolarize the membrane, and this is a decrease in membrane potential. And the way you need to think about potential is that it's a difference between the two. So if you decrease the difference, then the potential is getting smaller. So in terms of our neuron, the closer we get to 0 millivolts, the more we are decreasing the membrane potential or depolarizing the neuron. 0 millivolts means there is no difference between the interior and the exterior. Because potential, what we're measuring, is a measure of the difference. 0 would mean no difference. So if we were to look at this, here's our negative 70, here's 0. Depolarizing events are going to take us closer to 0. Our membrane potential, our voltage, is going to go closer to 0. Hyperpolarizing is the opposite. We would increase the membrane potential, meaning that the difference between the two gets larger. So here we start at negative 70. If we have some type of stimulus that drops us even further down, closer to a negative 100, that's going to be hyperpolarizing. So depolarizing means we're taking the change away. We're getting closer to zero. Membrane potential is decreasing. Hyperpolarizing, hyper means above, we are taking the difference and we are making it much larger, so we are increasing the potential. So anything that takes us closer to zero, depolarizing. Anything that takes us farther away is going to be hyperpolarizing. Now, graded potentials are local changes to the membrane potential that happen on the dendrites. So this is, remember, the input zone. So when two neurons are talking to each other and one of them releases a neurotransmitter, it's going to open up ligand-gated channels on the dendrites of the receiving neuron. So a few things about graded potentials. They decrease in magnitude as they travel further away from the channel. So essentially, you picture there's a bunch of ions flowing in right here. Those ions can only travel so far, so you're going to get a very local change. Here you can see it here. So if this is where we open it up, this area changes, but it can't spread too terribly far. Okay, we just don't have enough ions coming in to allow it to really truly travel. So these are short distance signals. The magnitude will vary with the stimulus. That's what we're talking about in terms of a graded response. It's going to vary depending on how many channels I open. And that is directly related to how many ligands I have. Now, a graded potential, a local potential, can be either depolarizing or hyperpolarizing. So it can go in either direction depending on what type of channel it's going to open. So we're going to take a second here and we are going to draw out what we've talked about so far before we move into action potentials. Okay, so we're going to draw out a neuron and kind of put together this stuff that we've talked about so far. All right, so we've got a cell body over here, and we've got an axon over here. We know that our primary intracellular cation is potassium, and our primary extracellular cation is sodium. Chloride always likes to hang out with sodium, so it's going to be in high concentration out there as well. And this is going to set up a situation where we're going to have a more negatively charged interior versus a more positively charged exterior. And when there's nothing going on in the cell, the difference between them is negative 70 millivolts. That's what's called resting membrane potential. So what we're saying is when we compare the two, when we compare the interior to the exterior, there is a difference that equals negative 70 millivolts worth of a potential to do work. This potential can change. It can go closer to zero. Zero would mean that there is no potential, that the two sides match, or the difference between them can get even bigger, and we can go closer to 100, negative 100 millivolts. So when we think about resting membrane potential, we say because there's a difference that the cell is polarized at this point. So remember that the word polar means different from one side to another, and when we talk about the difference between the negative and the positive in terms of the charge, we have a difference. The cell is polarized at resting membrane potential. Any sort of activity that brings the potential closer to zero is called depolarizing. So we are taking the polarization or the difference away because we're going closer to zero, and zero means no difference at all. If we make the difference bigger, anything that brings us in this direction, that's going to be called hyperpolarizing. So anything that creates a larger difference between the two would be a hyperpolarizing stimulus. Now, over here, I haven't drawn them in, but here's where our dendrites would be. And we know that our dendrites are our input zone. It's where we're going to have local potentials that use ligand-gated channels. So let's go ahead and put some ligand-gated channels in here. Let's see what would happen if we were to open them. So, we know that neurotransmitters are going to function as our ligands. So we have a neurotransmitter that's released onto our dendrite, and it opens up a sodium channel. So there's our ligand, and our sodium channel opens. The gradient for sodium is going to be to move into the cell. So the cell is going to gain positive charges. In terms of the effect on potential, it's going to be a depolarizing event. The interior is going to become more positive, which is going to bring it closer to this. So we are decreasing the difference between the two. So sodium channels, when they open, are going to be depolarizing. Now, if I happen to have a neurotransmitter that opens up a potassium channel, the gradient is going to be in the opposite direction. Potassium is going to leave the cell. When that happens, the already negative interior is losing positive charges. It is going to hyperpolarize us. We are going to end up moving in this direction, farther away from where we started. So anything that opens up a potassium channel falls under hyperpolarizing. If I have a neurotransmitter that opens up a chloride channel, chloride, we don't have a lot in here, so it's going to move in. Not quite to the level that sodium is because we do have a negative interior, but it will move in because of its concentration gradient. The negative interior is going to gain more negative charges. Chloride channels opening would fall under hyperpolarizing. So this is where we can get an idea of what's going on the dendrites and how the signal comes into the cell and what it does. Okay, so all of these signals are either going to depolarize or hyperpolarize. Now these are all local. They don't travel very far. There's not very much coming in. It's a small amount. But as you sum them up, there will be an overall effect on the potential. And if we get to what we call threshold, which is negative 55 millivolts, then we're going to move into action potential. Action potentials happen over on the axon, and they use voltage-gated channels. So we're no longer dealing with ligand-gated channels, we are dealing with voltage-gated channels. And the axon hillock area is absolutely packed with voltage-gated channels. And when they open, we're going to get this massive movement of sodium into the cell. It's going to be a huge amount of sodium coming into the cell, and that's going to start off our action potential. That's going to be a much more long distance signal, and it's going to be the signal that leads out of the neuron. So remember that we do have one-way flow. We've got information coming in on the dendrites using local potentials that utilize ligand-gated channels, and the information is going to leave from the axon using our voltage-gated channels. So make sure that this all kind of makes sense to you, that this is where we're starting before we jump into exactly what's happening with our action potentials. Okay, now that we've got all of that situated and we understand where our ligand-gated channels are, where voltage-gated channels are, how opening potassium, sodium, and chloride channels are going to affect potential, what threshold is, all of that stuff, now we're going to move into what happens with an action potential. So these are going to be long distance signals. They're going to originate at the axon hillock, and they're going to go all the way down the axon. These utilize voltage-gated channels. Therefore, the magnitude is not going to change at all. It's all or nothing. We either hit threshold and the voltage-gated channels open, or we do not at all, and there is no action potential. We're going to see that they do not decrease in magnitude. That's a good thing. Remember, the axon is really long. It could be really bad for us if they just, sort of petered out halfway down our leg and we never actually get to move our toes. Okay, so we have no change in the magnitude. And it's going to be a very predictable pattern of depolarization followed by repolarization. So we don't see the possibility of, well, maybe it's depolarizing, maybe it's hyperpolarizing. Every single action potential follows the exact same pattern. So we start at -70. Between -70 and threshold, all of this is driven by local potentials. So this is all driven by opening up ligand-gated channels and hoping that we get to threshold. If we do, we will get a very specific rise, depolarizing up to about +30, maybe +35, followed by a very specific period of repolarizing that's going to drop past threshold, and actually there's a small little area of hyperpolarizing, and then we end up back at resting membrane potential. Every single action potential looks exactly like this. There is no variation in them. This actually shows you in terms of time what it actually looks like. This is actually sort of stretched out so that you can actually see them a little bit clearer, the different things that are happening here. But you can see that actually the action potential, the depolarizing followed by repolarizing, happens very, very quickly. And then you've got a period of hyperpolarization until we get back to resting membrane potential. So this is what it really looks like. This is so that you can actually figure out what's going on. Okay, so we're going to walk through the events and then we're going to take a look at the picture here. So we've got two types of channels, all voltage gated. We've got sodium and potassium channels. The difference between them is sodium channels are going to open and close very fast. So it's as if their door opens very, very quickly. Potassium channels are going to open and close very, very slowly. Okay, so that's the major difference between them. In the resting state, so at -70, both of the channels are closed. If we hit threshold, -55, that's the trigger for the voltage-gated sodium and potassium channels to open. Sodium channels are going to open very, very quickly, and you're going to get a massive increase in sodium in the axon hillock area because we have somewhere in the neighborhood of 350 to 500 of these voltage-gated channels per square millimeter of your axon hillock area. That's a huge number of them. And remember, it's all or nothing. They all open at -55. So you're going to have a massive amount of sodium coming in. Now, the potassium channels are also working on opening, but they're very slow. It's going to take them a while to actually get open. So overall, what's happening is sodium is rushing in, and it's bringing the potential past 0 all the way up to positive 30 something. Now, once we get up there to positive 30, positive 35, somewhere in there, the sodium channels become inactivated. So no more sodium is able to come into the cell. And by this point, the potassium channels have finally opened. So the potassium now is going to start moving out of the cell down its gradient. So we had a bunch of sodium coming in during depolarization, and it got to the level of positive 30 and inactivated our channels, but the potassium channels finally opened, so now potassium is leaving the cell. And because we're losing a positive ion, our potential is dropping back down. You do get a brief period of hyperpolarization because those potassium channels are just as slow to close as they are to open. So we're going to have a period where we let out a little too much potassium. But we will rebound back to resting membrane potential. So at this point, the electrical conditions are reset. We are back to resting membrane potential. But the ions are on the wrong side of the membrane. We have a bunch of sodium inside and a bunch of potassium outside. That is where your sodium-potassium pump kicks in. It is going to reset the ionic conditions so that we can have another action potential following this one. So that's where we kick in with the sodium-potassium pumps, and we're going to take 3 sodiums out and bring 2 potassiums in for every turn of the pump. Now, it's all or nothing. We either hit threshold and we have this exact thing happen, or we do not hit threshold and nothing happens. Okay, let's take a look at it. So here we have our 2 channels. So we've got sodium and potassium. At resting membrane potential, both are closed. If we hit threshold, they both begin to open. Sodium is fully open immediately. Sodium is going to start flowing into the cell, and it's going to drive the potential up. So we're going to get to 0, we're going to go past 0, and we're going to end up all the way up here at 35. That is going to cause then our sodium channels to close up. They're going to be inactivated here. At this point, the potassium channels have finally fully opened, allowing potassium to move down its gradient. And because our interior is losing positively charged ions, we're going to see the potential begin to drop again. So this is the repolarizing phase. Once we have completely got back down, we've hyperpolarized, we're back to resting membrane potential, everything goes back to the resting state and we're back to being closed. But notice what the ion conditions look like. Here's where we started. Now we've got a lot of sodium inside and a lot of potassium outside. That's where your sodium-potassium pump kicks in and is going to take these ions and move them to the correct side of the membrane for us so that we can then do this again. So you want to think that depolarization is driven by sodium coming in. Repolarization is driven by potassium going out. So now we need to think about how it propagates. We start at the axon hillock, but we've got to get all the way down an axon, all the way to the end, which we know can be pretty far away. So here we are looking at the concept of the axon and kind of looking at how it travels. And what you see is a wave that runs down the axon. So here we're looking at recording right here at this point. So right now this is at resting membrane potential, and here comes the action potential behind it, and you can see that it's flipping our charges. Okay, so where the action potential is occurring, we've got a more positive interior. So that's going to come through, and when it hits our recording point, we see that the voltage is going to increase. Okay, now following it is your hyperpolarization. So this all occurs in a wave. So now we come back down and we're at hyperpolarizing, and that's where that's occurring here. and the action potential has moved on, and you can see that the area behind the hyperpolarization is ready to go again. It's at resting membrane potential. So you can picture that this is just going to keep going all the way down, and you're going to have a wave where you're at resting, then here comes the action potential, then here comes hyperpolarization, and then you're at resting again. And this is going to work its way all the way down the axon. So you're going to get a propagation that's going to occur. Now, there is a refractory period where we have to take into account how soon can the neuron have another action potential. So how quickly do those waves we just saw show up? So if we follow an action potential, here's our local potentials. We hit threshold, we go up, sodium's coming in, we come down, potassium's coming out. We have to get basically to resting membrane potential again before we get out of what's called the absolute refractory period. No matter what amount of stimulus you were to apply to the neuron during this period of time, you could not stimulate another action potential. This is because the first one's not done yet. So picture what's happening here. Here, sodium's coming in. Here, potassium's coming out. We're literally still in the middle of the original action potential, so we can't trigger another one. Now, during this period, where we're in our hyperpolarization, so remember our potassium channels are a little slow to close. During this period, we are in what's called the relative refractory period. We could stimulate another action potential from here, but it's going to take a bigger than normal stimulus. So in other words, it's going to take more than this. And we know that opening sodium channels are how we stimulate the neuron. So it means that we're going to need more sodium channels to open. And we know that that in turn means we're going to need more ligand. Okay, we're going to need more neurotransmitters coming in and opening up sodium channels to get us to threshold. You're basically just looking at the difference. So here, I got to go from here to here. But if I start down here, I go from here to here. Okay, so I need a larger than normal stimulus to trigger an action potential when I'm in the relative refractory period. And here you can kind of picture again this progression down the axon where I've got an action potential here followed by the refractory period followed by our excitable membrane again. And this whole wave basically flows down the axon. And any of this green area could have another action potential following it. So there definitely is a limit. Now, this whole thing only lasts a few milliseconds, so, it's not a significant limit, but there is one. So then that leads us to how quickly can we do this? How fast can we go? And that's going to depend on the axon. So the larger the axon is, the faster the conduction, and also the degree of myelination. So if we have a myelinated axon, it's going to allow it to go very fast due to something called saltatory conduction that we're going to take a look at. So basically group A fibers, they're very large, they're myelinated. We're talking somatic sensory, so those are those unipolar ones that we talked about, and motor neurons. These are the fastest. We've got signals traveling about 150 meters per second. Group B would be an intermediate, somewhere around 15 meters per second. And group C are your really small and completely non-myelinated. And even those were traveling a meter a second. So that's still pretty significant in terms of how quickly this happens. So here you can see what happens on a dendrite. Here's your stimulus, and remember that it is going to decrease as it travels because we just can't get a lot of movement within here. They can only diffuse so far. Now let's compare a dendrite to an unmyelinated axon. Here you have a stimulus, okay, and you're going to get diffusion of them. And then what happens on an axon is you run into another set of voltage gated channels. And so as the membrane potential changes, it's going to open up these guys. And then more sodium is going to come in. And the membrane potential is going to change, and it's going to open up these guys. And more sodium is going to come in. And then et cetera, et cetera, et cetera. That's how the signal doesn't change and it doesn't lose magnitude. You are opening up new sets of voltage gated channels all the way down the axon. So in the axon hillock area, you get this huge influx of sodium, and it's like hitting the first domino in a line. You're just going to keep depolarizing, and every time you hit threshold, a new set of channels is going to open, allowing even more sodium to come in. So that's how we have a long distance signal that can go from, the spinal cord all the way down your leg and under your foot to your toes and activate those muscles. It's because all along the length of the axon, we've got voltage gated channels that are being stimulated as soon as the membrane is depolarizing to threshold. So that's going to be fairly quick. If we add myelin into the situation, we're going to get even faster conduction because we're going to be able to skip huge portions of the membrane. We don't have to depolarize all of this. It's being insulated by the myelin. So we skip to the areas where the axon is exposed, and if you remember, this is the nodes of Ranvier. This is where we find the voltage-gated channels in a myelinated axon. They're not all along the length. We don't have to depolarize every single piece here. We're able to skip over huge portions and jump around. So picture this. You need to walk from here to the other end of whatever room you're in right now. And you've got two ways of doing it. The first is heel to toe. So you have to cover every piece of the floor. So you're going to take a step, put your heel down at your toe, take another step, heel down at the toe. It's going to take you a little ways. You've got to cover the whole floor. Or you can start where you're at and take the biggest steps possible. Huge steps. You're going to skip over huge parts of the floor. You're going to get over there a lot faster. That's the difference between these two. Here, we basically have to depolarize the entire axon. It's going to take us a while. That's like heel toeing all the way across the room. Here, because my voltage gated channels are only found in the nodes, we're going to be able to jump past huge parts of this. This jumping is called saltatory conduction. So you can see that the action potential appears to kind of jump from node to node as you're watching, because we just ignore all of this part, okay? We're able to basically just flip one node to the next, and that's like taking your really big steps to get you across your room. And so that's why our big motor neurons and our sensory neurons that are bringing information in are myelinated, because we need the fastest possible conduction there.